In the second part of our discussion of nervous tissue, we will discuss some features of nervous tissue and support cells. This continues our objective of describing the basic cells and functions of the nervous system. Myelination is an insulating layer around a nerve fiber. In the central nervous system, cells called oligodendrocytes do this, and in the peripheral nervous system, Schwann cells achieve this function. Myelination is formed from wrappings of the plasma membrane, and remember, the plasma membrane is composed of 20% protein and 80% lipid, which is why myelin looks white. All myelination is completed by late adolescence. In the peripheral nervous system, hundreds of layers wrap an axon. The outermost coil is the Schwann cell, or neurolemma. This is covered by a basal lamina and endoneurium. The initial segment is the area before the first Schwann cell, and in this initial segment, the axon hillock forms a trigger zone where signals begin in the neuron. Myelination begins during fetal development, but proceeds most rapidly in infancy. Oligodendrocytes myelinate several fibers in the central nervous system. Because these cells are in the central nervous system, there is no neurolemma or endoneurium. Those are only in the peripheral nervous system. The myelination spirals inward with new layers pushed under the old ones. Gaps between the myelin segments in either the central nervous system or the peripheral nervous system are called nodes of Ranvier or myelin sheath gaps. In the peripheral nervous system, Schwann cells envelop axons. The Schwann cell rotates around the axon, wrapping its plasma membrane around in successive layers to create the myelin sheath. We also have unmyelinated axons of the peripheral nervous system. These axons are often held by Schwann cells in grooves on their surface with only one membrane wrapping. So they do have a little bit of protection, but not the same level of myelination of the myelinated neurons. Axonal transport is the way that we move things from the axon terminal to the soma and vice versa. All of our proteins are made in the soma, so these have to somehow be transported to the axon and axon terminal. The reasons we might need these proteins in the axon are to repair the axolemma, or cell membrane, of the axon. We may need more gated ion channel proteins. We may need enzymes or neurotransmitters. Fast axonal transport goes either direction up to 400 millimeters per day. Anterograde is toward the axon terminals using a protein motor system called kinesin. Organelles, synaptic vesicles, and small molecules are carried through fast anterograde transport. Acetylcholinesterase, the enzyme that breaks down acetylcholine, is also carried via anterograde fast axonal transport. Retrograde fast axonal transport is going towards the soma using a protein motor system called dynin. Recycled materials and worn-out organelles are carried on this protein motor system. Some signaling molecules are also carried on these protein motor systems, and pathogens like viruses and the tetanus toxin can move through retrograde fast axonal transport. Slow axonal transport is only anterograde and moves 1 to 10 millimeters per day in a stop-and-go fashion. This moves new axoplasm for repair and regeneration, as well as new cytoskeletal parts to the axon. We have a number of neuroglial cells. Neuroglial cells are cells that support neurons. Astrocytes are delicate branched stars with gap junctions. They connect to capillaries and to neurons. Their functions are to contribute to the blood-brain barrier, regulate the composition of brain tissue fluid, including potassium and neurotransmitters in the extracellular fluid. They connect neurons to the blood supply to help feed that neuron, and they secrete nerve growth factor to promote synapse formation. They also can have an electrical influence on synaptic signaling. They, however, never actually carry the electrical impulses. The action potentials are always carried by neurons. We find astrocytes in the brain. Oligodendrocytes are a cell with many processes that wrap many axons in myelin and form the myelin sheath in the central nervous system. These cells are found in the central nervous system. Ependymal cells are epithelial cells that vary in shape from squamous to columnar and produce and circulate cerebrospinal fluid. We find these lining cavities in the central nervous system. Microglia are an immune cell that develop from macrophages and have lots of processes but are very small in size. They clean up damage and monitor neuron health. We find these near areas of infection or trauma in the central nervous system. In the peripheral nervous system, we have Schwann cells, which wrap around the axons of nerve fibers and myelinate these fibers in the peripheral nervous system. Satellite cells are also found in the peripheral nervous system, and they are found around cell bodies. They're flattened around the soma and help with signaling and feeding neurons. Here's an image depicting the different cell types, like the oligodendrocyte, the astrocyte, the microglial cell, and the ependymal cells that we find in the central nervous system. Here we have a diagram of Schwann cells and satellite cells around the cell body of a neuron in the peripheral nervous system. Next, we're going to look at how local potentials are formed in the neurons.